Hello there, everyone, and welcome back to another episode of Brain, Biology and Beyond. Today, we're going to be exploring a rapidly evolving, fascinating frontier in psychiatry and neuroscience, the link between neuroinflammation and mood disorders. Historically, the brain was thought of as immune privileged, largely disconnected from the body's immune system. Today, we know that this is not the case. Our goals of this session are threefold. First, we will establish a baseline by looking at how the immune system functions within a healthy brain. Secondly, we will be examining the mounting evidence linking neuroinflammation to mental health conditions such as depression, amongst others. Finally, we'll be discussing how biological insight translates into practical real world applications from cutting edge treatments to everyday lifestyle interventions. So strap yourselves in. To understand how the immune system affects our mood, first let's start with the universal human experience, catching the flu. We all know how miserable this feels. You experience profound fatigue, fever, aches, and a distinctly low mood. You don't want to socialize, you don't want to work, and you just want to curl up in bed. Crucially, these psychological changes are not an accidental byproduct of the virus. They are an evolutionary design known as sickness behaviors. Your immune system deliberately alters your behavior, forcing you to withdraw, conserve energy, and dedicate all bodily resources to fighting off the infection and recovery. Within the immune system, there are a number of different cellular players who are responsible for orchestrating this response. The immune system is a complex and highly coordinated network of cells and signaling pathways working tirelessly to defend us against pathogens. While we are familiar with peripheral immune actors such as B and T lymphocytes, which generate antibodies and targeted infected cells, and macrophages, which engulf cellular debris, the central nervous system has its own dedicated defense force in the brain. This role is taken up heavily by glial cells, including astrocytes and microglia, which we will focus on shortly. When the body detects a threat or tissue damage, it launches the inflammatory response. Think of this as the body's emergency broadcast system. It begins with the release of specialized proteins called cytokines, which act as chemical emergency signals. These cytokines rush to the scene and recruit other immune cells, hormones, and vital nutrients to the affected area to initiate repairs. In a healthy system, this response is tightly regulated. It ramps up to fight with danger, and once the problem is resolved, it shuts off, allowing the tissue to return to normal. Now let's have a look at some of the resident immune cells of the central nervous system, the microglia. They are the primary immune defense within that system, and you can think of them as a highly dynamic cleanup crew. They are constantly patrolling, clearing out cellular debris, defending against pathogens, and even pruning weak or unnecessary neuronal connections to optimize brain efficiency. Sometimes this is called pruning of synapses. However, they can change their shape and function depending on the environment. In mood disorders like major depression, we see a problematic shift. Microglia switch into a hyperactive pro-inflammatory state, often called the M1 phenotype, while simultaneously dialing down their protective anti-inflammatory M2 state. This chronic shift can alter brain chemistry and architecture, and also serves as a potential target for therapeutic intervention, but that we will get to later. As we move forward, we need to ask ourselves what happens when the cleanup crew doesn't turn off? How does a temporary protective biological response morph into a chronic state threatening our mental well-being? Well, let's break down the different timelines of inflammation to understand this shift. There are many, many diseases which have shifts in the inflammatory state involved, but we need to be able to differentiate between the two types of inflammation, acute and chronic. Acute inflammation is your friend, is an immediate short-term response driven by the activation of monocytes to heal a sudden injury or an infection, and it resolves quickly. Chronic inflammation, on the other hand, is a major health hazard. Here, the immune system gets stuck in the on position. Monocytes, macrophages, and other cells continuously pump out pro-inflammatory cytokines over months or years because the system fails to self-terminate. This prolonged immune fire begins to cause collateral damage, actively harming healthy brain tissues, leading to disorder. And it is found at the root of many different disorders. Regulating the inflammatory system is therefore extremely important. One of the main axes involved in this is the HPA axis, or the hypothalamic pituitary adrenal axis, which controls the stress response to inflammation in normal brains. And this also works in conjunction with glucocorticoid receptors. When stress hormones bind to the glucocorticoid receptors, they trigger a negative feedback loop that tells the body to calm down, exerting a powerful anti-inflammatory and immunosuppressive effect to keep the system from running wild. So let's have a look at those mechanics of that HPA axis in a little bit more detail. Whether you face a physical threat or a psychological stressor, the cascade starts in the paraventricular nucleus of the hypothalamus, which synthesizes corticotropin-releasing hormone, or CRH, along with vasopressin. These chemical signals to the pituitary gland to release ACTH into the bloodstream. The ACTH travels down to the adrenal glands sitting atop your kidneys, prompting them to secrete cortisol, our primary stress hormone. In a healthy loop, this circulating cortisol travels back up to the brain, binds to glucocorticoid receptors on the pituitary and hypothalamus, and effectively shuts off the further release of CRH and ACTH. It is an elegant, self-limiting loop that works well under normal conditions. Cortisol often gets a bad reputation as a toxic stress chemical, but is actually vital for our survival. As the final product of the HPA axis, its main job is to restore equilibrium to homeostasis after a shock to the system. Cortisol modulates the neuroendocrine and immune responses, helping allocate energy by regulating metabolism, managing inflammation, and stabilizing cardiovascular function during a crisis. Problems don't stem from cortisol itself, but rather than what happens when the brain stops responding to its signals, or a numbing of the glucocorticoid receptors. So when chronic stress breaks this regulatory system, we see distinct patterns of HPA axis alteration, which correlate with different psychiatric and neurological conditions. On the one side, we have hyperactivation, where the axis is constantly firing and overproducing stress hormones. This is frequently observed in classic major depression, schizophrenia, and Alzheimer's disease. On the flip side, we also see hypoactivation, where the system becomes exhausted and under-responds. This blunted response is characteristically seen in PTSD, chronic fatigue, and atypical depression. Both extremes represent a system which is profoundly out of balance and in need of restoration. So this brings us to a major paradox in psychiatric research, with individuals with major depression often showing incredibly high concentrations of cortisol in their blood, that they remain deeply depressed and inflamed. Why doesn't that cortisol shut the stress response off like it's supposed to? Well, the answer lies within the glucocorticoid receptor resistance. In roughly 80% of depressed patients, prolonged chronic stress has essentially fried the system, causing these receptors to become desensitized. It is highly analogous to insulin resistance in type 2 diabetes. Because the brain is blind to the cortisol signal, it cannot inhibit CRH and ACTH. The brakes are cut, the stress axis spins out of control, and the body is flooded with destructive pro-inflammatory cytokines while losing its protective anti-inflammatory defenses. The systemic inflammation doesn't just stop at the brain, it impacts the entire body, beautifully illustrating the link between depression and cardiovascular disease. High systemic inflammation triggers the liver to produce C-reactive protein, or CRP for short, which is a notorious clinical marker of cardiovascular damage. Patients with depression frequently have highly elevated CRP levels, drastically raising their risk for heart disease. Strikingly, this relationship is a two-way street. Having coronary heart disease significantly increases your risk of developing depression. Treat the heart and you may help the mind, and soothing the mind and you protect the heart. And this may well explain why those with chronic stress tend to have shorter life expectancies, among other reasons. So we're going to have a little look now at the bidirectional relationship between the blood-brain barrier and inflammation. Normally, the blood-brain barrier acts as a strict border control for molecules to enter the brain tissues. However, factors like chronic stress, a poor diet, and gut dysbiosis can degrade and weaken this barrier, making it leaky. This allows peripheral pro-inflammatory cytokines to leak directly into the brain. Once inside, these cytokines wreak havoc on neurons and neurotransmitter network through a myriad of mechanisms. Animal models confirm that an excess of these cytokines stunt neurogenesis, the birth of new brain cells, and triggers high levels of oxidative stress via reactive oxygen species. Ultimately, the combination of chronic stress, gut health issues, and genetic vulnerabilities lock the patient into a self-perpetuating cycle of neurological inflammation and mood decline. But that's not all. The inflammatory pathway can also affect neurotransmitter production themselves. For decades, psychiatry relied on the monoamine hypothesis to explain depression. The idea that depression is simply a chemical imbalance caused by low serotonin, dopamine, or norepinephrine. Today, we know that this is far too simplistic. Inflammation directly alters these neurotransmitters through an alternative biochemical pathway called the kynurenine pathway. Tryptophan is the amino acid our body uses to build serotonin. However, when inflammation is high, it activates enzymes that divert tryptophan away from serotonin production and push it down the kynurenine pathway instead. In a healthy state, This pathway is balanced and creates neuroprotective agents such as carnitinic acid. But under inflammatory conditions, the pathway skews heavily towards producing neurotoxic compounds, specifically quinolinic acid, which overactivates NMDA receptors and contributes to structural brain changes in depression. The NMDA receptor is involved in plasticity, so that's how it works pretty much. Overactivation is not necessarily a good thing. If depression is so debilitating, why hasn't evolution weeded it out? From an evolutionary perspective, the biological machinery underlying depression may have originally offered a survival advantage. In our ancestral past, predators and pathogens required an immediate and aggressive immune inflammatory response to survive. The problem is that our brain's threat detector, the amygdala, cannot tell the difference between a physical predator and a modern psychological stressor like financial strain or social rejection. It triggers the exact same fight or flight response, spiking your blood pressure, cortisol, and inflammation. Evolution favored an overreactive immune system because a false alarm is at least safe, but missing a real threat is fatal. Unfortunately, our modern world has changed, and we don't need that rapid response anymore, and biology lags behind that as if they were physical infections. And if we have a look at many of the diseases of the modern world, just on this slide here, I say of the modern world, many neurological diseases that are on the rise, we can see that Alzheimer's, multiple sclerosis, Parkinson's, traumatic brain injuries, major depression, amongst others, all have overlapping mechanisms at the root of their pathology, all with the associated with increased release of various pro-inflammatory cytokines such as interleukin 1 through 6 and 7, interferon gamma and TNF alpha, all of which can lead to symptomology. So there are some hypotheses that all of these different diseases are indeed just different versions of the same disease. This also may well explain why those people who may suffer mental health issues when they are younger, such as depression, anxiety, and even ADHD, may be at greater risk of Alzheimer's when they are older due to the overlapping heightened inflammatory risk. But now we've looked at these roots, we must ask the critical question: what can we actually do about it? So now we're going to pivot and explore how we can target these therapeutically. We will start our exploration of treatments by looking at different pharmacological interventions. So neuroinflammatory drugs, I've actually got a slide on neuroinflammatory drugs, but they have been found, neuroinflammatory targeting drugs have been found to be somewhat effective, and there are many different examples. These include microglial modulators. One of our most promising targets, in fact, is shifting their angry pro-inflammatory M1 microglia back into their helpful protective M2 state. Interestingly, some of the existing tools do this already, with SSRIs already having been shown to reduce inflammatory markers such as TNF alpha and nitrous oxide. Rapid-acting drugs such as ketamine also appear to exert their antidepressant effects, partly by shutting down microglial inflammation. Furthermore, we are looking at repurposed drugs like minocycline, an antibiotic that happens to be an excellent microglial inhibitor that lowers HPA axis and boosts BDNF, which is a key protein in brain neuroplasticity. Even natural compounds like plant-derived terpenoids found in fruit and vegetables are being studied for their ability to soothe microglial pathways and ameliorate depressive behaviors. Now, aside from those compounds, there are many different drugs that may well be used to actually directly target the cytokines themselves. This is where cytokine inhibitors come into play. They are monoclonal antibodies typically, which are used successfully in autoimmune diseases such as rheumatoid arthritis. And there have been clinical trials have shown that TNF inhibitors can improve recovery outcomes, and drugs that block interleukin, specifically interleukin 1 and interleukin 6, have been shown to significantly reduce depressive symptoms in highly inflamed patients. The catch is that these are heavy-duty systematic medications, meaning that they need to be carefully targeted to ensure that we know where they will benefit most and have the least side effects. There are also a number of genomic frontiers where, by utilizing cutting-edge tools like CRISPR, gene editing, or RNA interference, scientists are looking at ways to selectively knock down or silence specific genes responsible for overproducing pro-inflammatory cytokines within the brain, offering potential for long-term and targeted relief within those individuals, although this is still very much within its infancy. Alongside gene editing, Various fields such as stem cell therapy and regenerative medicine have been investigating the use of stem cells to introduce fresh, healthy cell lineages into the nervous system. These cells can release a steady stream of anti-inflammatory signals, helping to rebuild brain tissue damaged by years of chronic neuroinflammatory stress. Within mouse models, transplanting neurostem cells, neurological stem cells into Alzheimer's disease model mice improves their memory, increases synaptic density, and raises neurotransmitter levels of glutamate. Interestingly, while the cells survive long term and promote some cognitive recovery, it doesn't actually change the structural load of the tau pathologies, which are the different fibrils that build up within the brain, and therefore doesn't actually know if it's going to be, a reversal of that pathology or whether it's just a temporary boost. There are, as to date, clinical trials within people are yet to be successful, although again, this is a field within its infancy. So while high-tech drugs and gene therapies and stem cells are an exciting future with much research on the way, we have much more evidence-based lifestyle modifications that we can actually make now that can profoundly influence neuroinflammatory pathways. Let's have a look at the choices that we may make. So, the food we eat is either a fuel source for inflammation or a powerful medicine against it. Diets high in processed foods, saturated fats, and refined sugars break down the gut barriers and lead to systemic inflammation, and lead to increased oxidative stress, damage the brain, and cognitive dysfunction. Conversely, anti-inflammatory diets, which have rich levels of omega-3, high levels of fruit and vegetables or polyphenols, and fermented foods, act as a shield for the brain, lowering that inflammatory cytokine profile through a myriad of mechanisms. They may well improve the gut microbiome, reinforce the blood-brain barrier, and activate internal antioxidants, increasing BDNF to support neuroplasticity and mood. Now, movement is becoming increasingly recognized as a prescriptive drug, or as good as prescriptive drugs for treating depression. Indeed. So, if we're looking at large-scale studies that confirm regular activity, obviously prevents major depressive episodes, and it does so by, in part, reducing the neuroinflammatory profile. Different studies report different things, with different types of exercise being found to exert slightly different profiles or slightly different effects on the inflammatory profiles, with what's recommended is about 150 minutes of cardiovascular exercise per week of some description of, say, zone 2, as well, alongside strength training to get the benefits from that. Although it is interesting to note that overtraining can affect depressive symptoms and lead to altered stress and overtraining syndrome, with athletes who overtrain being at higher risk of burnout. Combinatorial modalities of them both seem to be the most effective, as mentioned. If I move on to the next part, we can have a look at mindfulness. So, meditation, which is an ancient practice, has come into a bit of a renaissance within these recent years, with a lot of research now starting to back it up as a potential intervention or prevention for neuroinflammatory disorders, including mood disorders, amongst others. In addition, it seems to have a role on curbing inflammaging, a chronic low-grade inflammation which accelerates with stress and aging. Studies show that regular mindfulness can reduce oxidative stress, with enzymes in the mitochondria being reduced, such as oxidative phosphorylation complex number 1. And also this leads to reductions within the telomere shortening process, which is a hallmark of aging. It seems to sustain its lowering of inflammatory markers over time within TNF alpha, although some markers such as interleukin 6 and CRP show a delayed response. So to wrap it up, in today's session, I'm going to talk about where the scientific landscape might be heading. The field of neuroinflammation is experiencing an explosion within innovation at the moment, and there's far too much for me to talk about for this video to be, relatively short. So we're going to have a look at a few of the avenues where there is some excitement, including the improvement of organoid modelling for the CNS, improved biomarker panels, and nanoparticles to deliver neuroinflammatory drugs and tune the glial response. So, first big leap is how we are going to be modelling the human brain. Traditionally, we have relied heavily on mouse models, which frequently fail to translate cleanly into human biology due to genetic differences and physiological differences. Today, stem cell technology allows us to grow three-dimensional, simplified models of the human brain in a dish known as organoids, developed from patient-derived stem cells. Early brain organoids had a few immune cells within them, and thus may not have actually represented the brain in its entirety, given how important the neuroinflammation and the inflammatory response is within the brain. Recently, pioneering breakthroughs in cold culturing have successfully introduced functional microglia into these 3D models. In fact, a landmark study by Schaffer and colleagues within 2023 successfully incorporated glial cells within a model of the brain of derived from stem cells from a patient with autism, and this highlights the future of developments within the field and potentially enables us to better target neuroinflammatory pathways by understanding how they work within the tissues rather than relying on mouse models. The second massive frontier to address is the drug delivery obstacle. Now, the blood-brain barrier is relatively impermeable, with many excellent inflammatory drugs fail simply because they cannot cross this border control. Enter nanotechnology. Engineered nanoparticles designed with precise chemical properties and sizes that can seamlessly cross the blood-brain barrier. Already demonstrating immense promise within models of Alzheimer's, Parkinson's, these nanoparticles can carry a therapeutic payload directly to overactive immune cells. Once inside, they can suppress the release of toxic cytokines and actively switch the cells, the glia in particular, from M1 to M2, which is beneficial and anti-inflammatory. However, nanotechnology can be thought of as a bit of a double-edged sword because these materials are incredibly small and they are highly reactive, and their unique physical properties can occasionally backfire. If not engineered perfectly, certain nanoparticles can actually trigger the exact neuroinflammation that we want to prevent. For instance, experimental studies have found that exposure to silver nanoparticles cause glial cells to mistakenly dump out nitric oxide, hydrogen peroxide, triggering oxidative stress. They can even accelerate interleukin-1 release and cause toxic amyloid beta protein clumping, mirroring the pathology of Alzheimer's disease. To harness this technology safely, researchers are optimizing safe dosing strategies and utilizing protective coating processes like pegylation, which is the addition of polyethylene glycate to help minimize toxicity. If we can master the balance, then we can hold the key to directly tuning the brain's immune system through precision medicine, changing the future of neuroinflammation treatments, including Alzheimer's and depression, forever. Thank you very much for watching. I'll be putting some references in the description of this video, and if you are interested on anything within neuroscience, then please do check out the rest of the videos on my channel. I look forward to speaking to you again. Thank you.